Hello, Amin and I are presenting the Taurus One study today, which examines the safety of magnetic brain stimulation for people living with multiple sclerosis. So first, what is myelin and what does myelin actually do? So the yellow cell here is a neuron that is coated with myelin, which is in green. So myelin is an insulating layer that wraps around the neurons and protects it. Myelin is very important because it allows the electrical signals to travel along the neurons quickly and reliably. However, in multiple sclerosis, the immune cells, which is in pink color, enters the brain and attack myelin. This results in myelin loss. This also affects the electrical signals don't reach where they need to go. And ultimately, people end up with coordination problems or cognitive problems or other problems associated with multiple sclerosis. So, if we can find a way, a treatment to add myelin back to the brain, we can probably reverse the damage from MS. So that's where we come to the idea of myelin repair therapy. So people living with multiple sclerosis have also identified that myelin repair is one of the biggest unmet needs because currently there is no treatment to promote remyelination. So that is why our team is currently trying to develop a treatment for myelin repair. And one promising approach that we are using is magnetic brain stimulation. So what is magnetic brain stimulation, MBS for short? So MBS involves holding a copper coil over the head, and then within the coil, there is an electric current running through it, which then generates magnetic field. So this magnetic field can further induce another electric current running in the brain tissue underneath the coil. And this induced electric current can change the electrical signals where the cells used to communicate. So this is the first time that our team is using MBS as a remyelination therapy. So how might it work? So the white cell here is the cell that can make myelin. So when myelin damage occurs, myelin is lost and that cell is also lost as well. Yep. And then the immature cells can respond to it. They can migrate towards the injury site. However, they can't make myelin. So this is where the MBS comes in. The MBS increases the cell activity. So the immature cells can mature into the myelinating cells. So they can add more myelin to the brain. So these laboratory findings that help us to move into the clinical stage involving people with MS. So in 2021, we completed the Taurus One study with a small number of research participants. And the primary objective was to determine whether MBS is safe and tolerable for people living with MS. We also obtained the preliminary data about the effects of MBS on the MS symptom severity, such as fatigue, depression, anxiety, quality of life, and also obtained the preliminary data on the myelin repair based on MRI scanning as well. So the Taurus One Trial was conducted at the clinical research facility at the Medical Science Precinct in Hobart in Tasmania. So 20 people living with MS were recruited in the Taurus One Trial, and they were randomized into either MBS group or placebo group. So in placebo group, the participants underwent the same procedure, but no real stimulation was applied. Before the treatment, participants completed the MRI scanning and also the questionnaires on the MS symptoms. After that, the participants attended 20 daily sessions from Monday to Friday over 5 weeks, and each session lasts around 15 minutes. Then, right after the treatment, participants again completed one MRI scan and the surveys on the MS symptoms again. And 3 months post the treatment, post the last treatment, participants were asked again to complete the surveys. And now Amin will continue to talk about the results of the Taurus.1.
Hello everyone and good afternoon. I hope the lunch was good. So I'm going to talk about the results, including the adverse events, which is our first and primary aim of the study. So as you can see in this table, the baseline characteristic of the participants, 20 participants who participated in this study, in terms of the sex and duration of MS diagnosis, which in terms of the statistical calculation is no different between these two groups. And in terms of the level of disability is measured by a standard measure of disability in people living with MS, which is EDSS. They were slightly different between the MBS group and placebo group. And this was sourced from the mild disability, the EDSS between 2 to 3.5, which in MBS group has more people assigned to this level. But as this is not the main exposure of this study, this slight difference is not going to make any difference in our study outcome. In terms of the MS time, they're fairly similar apart from the primary progressive MS that we had 4 participants in the MBS group. And this is the main and primary result of our study, which is the safety outcome. As you know, an adverse event is any untoward medical occurrence that may happen in any research study in participants who receive the treatment, which can be any intervention like medication or what we have here, magnetic brain stimulation. But these adverse events are not necessarily related to the treatment, and it could include even accident, getting COVID or flu-like symptoms, or the adverse events that we were looking for, such as headache, seizure, that as part of the defined adverse events related to MBS. But unfortunately, none of the study-related adverse events, which we call adverse reaction, none of the participants reported in this study. But we have several adverse events, including fractured arm post-fall, shingles, worsening of memory and balance, and generalized rash, which based on the neurologists in the study, neurologist decision, none of them were related to the intervention. As Tram said, we also had some secondary aims in this study, including a series of questionnaires, surveys, and also we used a mobile application called Floodlight Open to assess the cognitive function in participants in this study. I brought here one of the results from the Floodlight test, which shows the number of steps before, after 20 sessions and after 3 months. Although the differences was not significant, but as you can see, in blue color shows the MBS group, which fairly kept the level of baseline or before treatment number of steps during the study, but the placebo group slightly deteriorated the number of steps. These are preliminary results. We are not saying that our treatment 100% effective. This is an important thing to be noticed here. But these are one of the things that we are going to look further in the next stage. In terms of depression and anxiety symptoms, we use the scale to measure the severity of symptoms reported by the participants. And another important point here is that all these are the average symptoms or average severity in each group. In none of the groups, the average is above the standard or clinically significant depression or anxiety. So these are within the normal range that even myself, which I'm not clinically diagnosed with depression, might report these level of symptoms. And these two groups were not significant in terms of comparison of the level of anxiety and depression. Another scale was the Fixability Scale, which again, both groups reported the same level of fatigue symptoms during the study and 3 months follow-up with no difference, and the same as quality of life. Now we are going to the most important secondary aim of the study, which are the MRI outcomes. So as you know, MRI is an important indicator of disease activity in MS and play a role in diagnosis of MS. And MRI scans shed light to what happened in the brain during the MS pathology. In our study, between the two groups of MDS and placebo, none of the participants had any new or enlarging lesions between the beginning and at the end of the trial. In terms of brain atrophy analysis, as you know, brain atrophy is a natural process of aging, but in certain medical conditions, the most famous one is dementia, and also in MS, we can see accelerated and early onset of brain atrophy. Again, all the participants in our study had less than 2% change. This is the change before and after the treatment in brain volume between baseline and follow-up MRI. And there was no difference between two groups. But as Tram clearly described, the process of multiple sclerosis affect the white matter or myelin content of the brain. We were specifically interested in seeing what changes that this treatment, magnetic linear stimulation, would affect the myelin content of brain. QT value is inversely correlated with the myelin content in the white matter. And if you remember the picture that Tram shows, the device placed in the frontal side of the brain, which is called frontal lobe. So the myelin content in the frontal lobe is specifically very important for us to see whether is there any change or not. So as I said, this is a preliminary result. We didn't see any significant difference between two groups, but in placebo group, in gray color, the placebo group continuously lost the myelin content. So, but in the intervention, in the treatment group or MPS group, there was no change, and it remained stable. The myelin content in time 1, which is the pre-treatment, and time 2 after the end of the treatment. And in other words, the placebo group continued to lose myelin over the course of the study, while the MPS group maintained myelin at the baseline level. And these are the list of lovely group of project contributors who worked from the conception of the study and during the study. And especially on behalf of our team, I would like to appreciate all the participants who kindly contributed their time to this study, and our project funders, the list are here. Thank you for the event contributors today and the funders of this event, and thank you so much for your attention.